Hi everyone, welcome to the joy of botany, especially plant physiology. Let me introduce myself. I'm Kiran. I'm a 5-year integrated BSMS student of ISO Thiruvananthapuram. I found this is a great opportunity to talk to you about some concepts in the plant physiology. Thanks to you guys for that. Without a further ado, let's begin. Today I am going to emphasize on the topic importance of water, water potential, its components, and significance. Before entering into the topic, let us first understand what actually water is for a living organism. Earth is the only planet in the universe where life is possible because of the availability of water and oxygen. Water is an essential element for life. More than 80% of the fresh weight of cell and tissues are water. It has many uses for organisms, such as it acts as a solvent, as a temperature buffer, as a metabolite, also as a living environment. In case of plants, water helps in transporting important nutrients within the plant. So in today's topic, we will see the importance of water in plants, the physical and chemical properties of water which make it so important. We will try to understand physical forces that influence water movement at the cellular level, correlate the physical forces with water potential, and finally, we will see the pathway of water within the plant. So, the importance of water in plants. Water is a major constituent of protoplasm in cells. Water is the solvent. Mineral nutrients dissolve in it and enter a plant from the soil, as well as they are transported from one part of the cell to another. Water is a medium of metabolic reactions, as well as it participates in various reactions like hydrolysis, respiration, etc. Water imparts turgidity. It provides mechanical support and rigidity to non-lignified plant cells. The elongation phase of cell growth depends on the absorption of water. Different types of movements in plants are regulated by water, like nocturnal folding of leaflets of certain plants, swelling and shrinking of guard cells, which led to the opening and closing of stomata, etc. Now, let us talk about the properties of water that makes them so essential. First, we will consider the physical properties. Water is a liquid at physiological temperature, that is between 0 degrees centigrade and 100 degrees centigrade. It means water has a high boiling point and a high melting point. When we compare this to other similar sized molecules like carbon dioxide, ammonia, hydrogen sulfide, all these molecules are gases at room temperature. So, a mere change in temperature will not change the state of water. Thus, water is a very stable compound. Water has a high heat of vaporization. Water needs energy of about 44 kilojoule to convert it from a liquid to a gas. This property is responsible for water's use in evaporative cooling purposes. Water has a high specific heat, that is heat capacity. Water takes 4.184 joule of energy to raise the temperature of 1 gram of water to 1 degree centigrade. Thus, water is slow to heat up and cool down. Or, in other way, water resists temperature change. This property is important in water's role as a thermal buffer. Water has a high heat of fusion. This means water resists freezing. Energy that is required to break the collective hydrogen bonds holding water in a solid configuration. Conversely, a lot of energy, say about 6 kJ per mole, must be released by the water to freeze. This property is used by citrus growers. Prior to a light freeze, they spray fruits with water. Ice worms release the heat of fusion, which will help to protect the crop from serious cold damages. Water has a high surface tension. Water molecules at the surface are attached to each other within the liquid. This gives them a cohesive force, and as a result, the water can form a column in a capillary like xylem. The density of water decreases on crystallization. This occurs because when ice forms, each water molecule is hydrogen bonded to exactly four others. At 4 degrees, water is its densest, and each water molecule is attached to slightly more than four others, so it floats in water. Water has a high tensile strength and incompressibility. It is not possible to push the pistons together if water is put in a tube and on either end of the tube pistons are placed. Thus, water is a good hydraulic system because when it is squeezed, it doesn't compress and produce positive pressures. This pressure provides the driving force for cell growth and other plant movements. In a similar fashion, if the tube is filled with water and air bubbles are removed from them, pulling the piston away from one another will not break the water column. This will result in a suction. Negative pressure can develop in the water column and very sizable tension can be generated in a thin water column. Water is transparent to light. This is important because chloroplasts are obviously surrounded by water. If water was opaque, plants couldn't photosynthesize. Now let's talk about the chemical properties of water. Water is a universal solvent. Water dissolves more different kinds of molecules than any other solvent. Water is chemically inert. It doesn't react unless it is enzymatically designed to do so. Water dissociates into protons and hydroxyl ions. This serves as a basis for the pH system. Water affects the shape, stability, and properties of biological molecules because water forms hydrogen shell around them. Now, to maintain the water balance, plants depends on some physiological factors that regulate the movement of water within the plant. They are water currents, electroosmosis, suction force of the cell, which is also called DPD, diffusion, osmosis, and imbibition. Now, let us talk about each of these factors. First, water current. It represents the total movement of water based on its free energy. Electroosmosis. Electroosmosis is the movement of liquid through the pores of a membrane under the influence of an electric field. This electric field is generated due to the pectin and other substances present in the cell wall. They contain carboxyl groups dissociates into COO- and H plus ions, which give a negative and positive charges to the cell wall. Imbibition. It contributes to water penetration through macromolecules of protein colloids and cellulose pectin microfibrils. Imbibition is based on the colloidal and capillary effects and causes an irreversible increase in volume and mass. Now, about diffusion. The movement of particles or molecules from a region of higher concentration to a region of lower concentration is called diffusion. Simple diffusion play a very important role in the life of plant. Firstly, diffusion helps in passive salt uptake. The ions are absorbed due to diffusion. It is an essential step in the exchange of gases during respiration and photosynthesis. During transpiration, water vapor diffuses from the intercellular spaces into the outer atmosphere through the open stomata. The diffusing particles have a certain pressure called diffusion pressure, and the diffusion pressure is directly proportional to the number or concentration of the diffusing particles. Therefore, the diffusion takes place always from a region of higher diffusion pressure to a region of lower diffusion pressure, that is, along a pressure gradient. Now, osmosis. It is derived from a Latin word called osmos. Osmos means impulse or urge. It represents the movement of water or solvent molecules through a selectively permeable membrane. The direction of this movement is from a lower concentration to a higher one. As a result of the separation of two solutions by a semi-permeable membrane, a pressure is developed in the solution due to the presence of dissolved solutes in it. This is called osmotic pressure. Osmotic pressure is measured in terms of atmosphere. It is directly proportional to the concentration of dissolved solutes in the solution. More concentrated solution has higher osmotic pressure. To understand the movement of water through the cell, we have to consider cell as an osmotic system. The ability of an extracellular solution to make water move in or out of a cell by osmosis is known as tonicity. A solution's tonicity is related to its osmolarity. Osmolarity is the total concentration of all the solutes in a solution. A solution with low osmolarity has fewer solute particles per liter of a solution, while a solution with high osmolarity has more solute per liter of a solution. When solutions of different osmolarities are separated by a membrane permeable to water but not to the solute, water will move from the side with lower osmolarity to the side with higher osmolarity. Next, let me introduce you to these three terms: hypotonic, hypertonic, and isotonic. These are used to compare the osmolarity of the cells to the osmolarity of extracellular fluid around it. If the extracellular fluid has lower osmolarity than the fluid inside the cell, it is said as hypotonic. Hyper means less than to the cell, and the net flow of water will be into the cell. As a result, the cell swells. The cells are restrained from swelling by inwardly exerted cell wall pressure. The water pressure exerted by the cell contents against the cell wall is called the turgor pressure. Turgor pressure is the actual pressure exerted by the protoplast of the sturgid cell against the cell wall. In the reverse case, if the extracellular fluid has higher osmolarity than the cell cytoplasm, this is said to be hypertonic. Hyper means greater than to the cell, and water will move out of the cell to the region of higher solute concentration. This makes the cell flaccid. In an isotonic solution, the extracellular fluid has the same osmolarity as the cell, and there will be no net movement of water in or out of the cell. Here, iso means the same. Cell as an osmotic system. Now, in this figure, it is depicted that different pressure acting on the cell in different ways. When osmotic pressure or OP of the cell is low, in comparison to the adjoining cell, water moves out of the cell. In the reverse condition, when osmotic pressure is high, water enters into the cell. Entry of the water increases the turgor pressure of the cell. This pressure is resisted by the wall pressure. So we can say that wall pressure is equal to the negative turgor pressure. The amount of cell expansion is evidently determined by two factors: the osmotic concentration of the cell sap and the resistance of the wall to stretching due to its turgor pressure. This ability of the cell to absorb water is called suction pressure or diffusion pressure deficit. Mathematically, it is represented as diffusion pressure deficit, or dPd, is equal to the osmotic pressure minus the turgor pressure. The water potential. It is a modern term coined by Slater and Taylor in 1960, which is equivalent to diffusion pressure deficit. It is defined as the difference between the chemical potential of water in a biological system and the chemical potential of the pure water at the same temperature and pressure. It is manifested as a force acting on water molecules in a solution separated from pure water by a membrane that is permeable to water molecules. It is symbolized by psi W. It has a negative value because water potential is reduction in free energy of solvent in a system over its pure state. Water is absorbed by a system with lower psi W from another system with higher psi W. The components of water potential. Many different potentials affect the total water potential, and some of these potentials determine the overall water potential and the direction of the water flow. The relationship between water potential and different potentials are shown by psi W is equal to psi S plus psi P plus psi G plus psi M, where psi W is the water potential, psi S is the solute potential, psi P is the pressure potential, psi G gravity potential, and psi M is the potential due to matrix. Now let us have a closer look at each of these terms. First, solute potential. The relationship of solute concentration to solute potential is given by the Van Hoff equation, which is psi S is equal to minus MIRT, where M is the concentration in molarity of the solute, I is the Van Hoff factor, R is the ideal gas constant, and T is the absolute temperature. Pure water is usually defined as having a solute potential of 0. Where a solute is dissolved in water, water molecules are less likely to diffuse away via osmosis than when there is no solute. A solution will have a lower and hence more negative water potential than that of pure water. Therefore, the more solute molecules present, the more negative the solute potential is. Solute potential has important implications for many living organisms. If a living cell with a smaller solute concentration is surrounded by a more concentrated solution, the cell will tend to lose water to more negative water potential of the surrounding environment. Now, let us look at what pressure potential is. Pressure potential is based on mechanical pressure and it is an important component of the total water potential within the plant cell. Pressure potential is increased as water enters the cell. As water passes through the cell wall and cell membrane, it increases the total amount of water present inside the cell, which exerts an outward pressure that is retained by the structural rigidity of the cell wall. When referring to the positive hydrostatic pressure within the cells, Xi is usually called turgor pressure. Now, the gravity potential. Gravity causes water to move downward. Unless the force of gravity is opposed by an equal and opposite force, the potential for water movement thus depends on height. The effect of gravity on water potential Ψg depends on the height h of the water above the reference state, the density of water ρw, and acceleration due to gravity g. We write the relationship as follows. Xg is equal to ρwgh. Xg may have significant effect on water potential if vertical distance or height is considered, but at the cellular level, Xg is insignificant in comparison to Xs and Xp, and therefore can be omitted from the equation. Now, metric potential. When water is in contact with solid particles such as clay or sand particles within the soil, and cell walls, proteins, and other hydrophilic cellular materials inside the cell, the adhesive intermolecular forces between the water and the solid can be large and important. Within a tissue, since all cells have the same metric potential, so it can be omitted. So, the equation for water potential can be simplified as follows. Xi W is equal to Xi S plus Xi P, where Xi S has a negative value while Xi P has a positive value. The pathway of water uptake. Now, let us understand how water moves in the root. In the soil, water is transported predominantly by bulk flow, which is also known as diffusion. However, when water comes in contact with root surface, the nature of the water transport becomes more complex. From the epidermis to the endodermis of the root, there are three pathways through which water can flow. The three pathways are apoplast, transmembrane, and symplast pathways. In the apoplast pathway, water moves exclusively through cell wall without crossing any membrane. The apoplast is the continuous system of cell wall and intercellular airspace in the plant tissue. Here, the movement of water is by simple diffusion. The transmembrane pathway is the route followed by water that subsequently enters a cell on one side, exit the cell on the other side, next they enters the next series and so on. In this pathway, water crosses at least two membranes for each cell in its pith. Transport across the tonoplast may also be involved. In the simplest pathway, water travels from one cell to another via the plasmodesmata. The simplest consists of an entire network of cell cytoplasm interconnected by plasmodesmata in transmembrane pathways and simplest pathway. Water moves from cell to cell due to the difference in the solute potential. At the endodermis, water movement through the apoplast pathway is obstructed due to the Casparian stripes. The Casparian stripe is a band of radial cell wall in the endodermis that is impregnated with the wax-like hydrophobic substances like suberin. And the suberin acts as a barrier to water and solute movement. The Casparian stripes breaks the continuity of the apoplast pathway and forces water and solutes to cross the endodermis by passing through the plasma membrane. Thus, despite the importance of the apoplast pathway in the root cortex and the stele, water movement across the endodermis occurs through the symplast. Finally, all the solute, along with the water, reaches the xylem and through the xylem, water finally transported to the leaves and other parts of the plant. Finally, let us summarize all the concepts that we discussed in this lecture. First, we observed what are the properties of water that helps the plant or the living organism to sustain. Second, we came to know about the turgor pressure, osmotic pressure, and the diffusion pressure deficit, as well as the different osmolarities, hypotonic, hypertonic, and isotonic solutions. Then we observe the pathways such as apoplast, symplast, and transmembrane pathways through which water enters into the plant and ultimately reaches to the different parts of the plant. That's all for today's lecture. We will definitely continue with very interesting and exciting concepts in the topic plant physiology. I wish you a good day. Thank you.